Good morning, students. Today we will cover the second part um, of the clinical trials. In the last class, we discussed what clinical trials are, what are the different types of clinical trials, um, and so before going into the details, I would like to give you a I would like you to give you a thought about again that who sponsors clinical trials. So usually the profit-making companies usually sponsors the clinical trials. And why is it important to test the drugs or the treatments or the devices on animal cells or subjects before you test them onto the human beings? Like we studied in the last class about the preclinical trials. Preclinical means before the clinical trial starts, and these are usually done on the animals in order to check the safety and uh, the efficacy and to study the molecular mechanisms. So these are tested initially on either the animal cells and then um, onto the animals before we test onto the humans. Uh, so the clinical trials are done in different phases. So in the phase one, the researchers initially test the drug which they want to test onto a small group of patients or the people. So these are about 20 to 80 people, and this is for the first time they are testing. So these people on whom we are testing are the healthy patients or the healthy people. They never had any problem or no disease condition. They just want to test uh, this drug onto these people because they want to evaluate the safety of this drug and to identify if it has any side effects. So and if it is for veterinary animals, it is conducted onto the animals directly as such. So where do these people come from? So we want to recruit the peoples, the healthy volunteers we call, and where do they come from? So these clinical trials, like I said, they require these volunteers or the healthy people whom it has to be tested, and usually in many countries they are often paid. So the hospitals or the clinical trial agencies, they basically recruit these initial 20 to 50 patients, people, healthy people, and they pay them in order to undergo the phase one clinical trial. Many people would volunteer to become subjects in this clinical trial, and the question arises: what if it meant you receive a treatment for a disease that wasn't available to anyone else? For example, the COVID-19, it was never available before, but now it is available, and so the people who will recruit in them as a volunteers in this study, uh, they will be receiving a treatment for the first time for a disease which was not available early on. So there is a famous example before, but by Merck, that they tested HIV vaccine onto the initial healthy volunteers, and unfortunately, these healthy volunteers turned out to be infected with HIV when these vaccines were tried onto them, and then this company had to pay them millions of dollars for um, because these were healthy volunteers. What are the phase two clinical trials? So then after the phase one, the next comes phase two. So herein the experimental drug or treatment is administered to a large group of people. So that means it's not tens, but it is in hundreds in number. So this large group of people or animals are about 100 to about 300 to 500 in number, and the whole purpose of doing this phase 2 clinical trial is to determine the effectiveness. How effective is this drug? So the and to further see the safety of this drug in a large number of people. So the next comes the phase 3. So in here, the experimental drug is administered in thousands. So that means there may be about 3,000 to 5,000 at the max, which may be enrolled in this study. And this is to confirm its effectiveness and to check if there are any side effects, and to compare this drug with already existing drug. Maybe there is already an existing drug and you want to prove that your drug, the new experimental drug, is more effective as compared to the standard or the available drug, equivalent drug treatment. So so there are various concepts now which are associated with these clinical trials. So in many studies, the new drug is compared to placebo. What is this placebo? So the placebo is a product which is which looks like the new drug, but it doesn't have the active ingredient in it. Maybe if you look at a capsule, inside the capsule there will be white powder, which contains the active ingredient. But in this placebo, there will be white powder, but doesn't have any active ingredient. So people do not know that they are getting a placebo. They think that they are getting a equivalent capsule. So sometimes the test also compares with the new treatment against the existing treatment. So like I said before, if there is an existing drug which is available, so the purpose is also to showcase that this drug is better than the existing drug. So for doing this, people conduct blinded and double-blinded trials. So what are these double-blind and blinded trials? So a blinded trial is a trial in which the patient do not know if they are receiving the treatment or the placebo. So in this blind trial, so the patient is completely blinded. He doesn't know if he or she is receiving the treatment or the placebo with active ingredient or without active ingredient. So they're just getting the pill, for example. So in a double-blinded trial, in which both the patient and the person who is testing, both the researcher and the patient, they both don't know who's receiving the treatment. Because these trials, the blinded and the double-blinded trials, are done so in order to avoid any biasness. Maybe if you are the researcher and you may be biased to a healthy person, and you may give the drug to a healthy person rather than a sick person. So yeah, in order to avoid the biasness, these kinds of blinded and double-blinded trials are done. So why are these good ideas? What is the reason? Like I said, to avoid biasness and to include more of a randomization. So the randomization is nothing but it's a process by which the patients are assigned a group for the clinical trial. So they are assigned to a group, and these groups are assigned randomly. So one is for the treatment and one is a control, for example. So not purposefully. So you are not biased, like I said. So some people will receive the new treatment, some people may receive the already approved treatment or the existing drug which is in the market, and some people may receive the placebo, which doesn't contain any active ingredient or the drug, only the dummy drug. So if one treatment is found superior, the trial is stopped, and the trial is stopped, so the fewest patient will receive the less beneficial. So here we stop because you do not want to give the less beneficial treatment, and you want to continue with the more beneficial treatment. So this at this stage the trial is stopped, so that the fewer patients will receive the more beneficial treatment. So once this drug is proved satisfactory, so if this drug is really good and the trial is stopped, so the results are combined. So these results are all put together into a book or a comprehensive description, which means the methods, the results, of the human and the animal studies, how this drug was manufactured, what were the formulations which were done, and what is the shelf life or the IC50 value of this drug. So this collection is the information which is provided to the regulatory agencies, and we call it regulatory submission. And this is provided for review to the FDA, the US Food and Drug Administration, so that they can grant the sponsor approval to market this drug or the device or the treatment, because they think that this drug is more effective. So what are the results? So people test about 5,000 to 1,000 or 10,000 compounds and in the early preclinical stages, and only one of this 10,000 compounds goes for preclinical testing, and only one drug is approved for marketing. So the cost of failures has to be borne by the price of one successful molecule. So whatever 9,999 molecules which failed during this trial, so this one drug has to bear the cost of failures of all those drugs. Further, there is a phase 4 trial as well. So what is this phase 4 trial? So after a drug is licensed and approved by the FDA for the treatment is launched, so the researchers, they keep tracking its safety, because we do not know in the long run what this drug does. So seeking more information about the drug and its potential risks, benefits, and optimal use. So the long-term studies, they involve large group of participants, which are designed to reveal if there is any unexpected, maybe if you take a drug for a long duration, after 2 years somebody develop a cardiac problems, or they have a vision related problem, for example, which could be side effects, and small percentage of the individuals. So what are the timelines? So if you look, the preclinical trials runs for about 4.5 to 5 years. Phase 1 to 3 trials goes for 8.5 years. FDA approval takes 1.5 years, so it is about 15 years here. And the phase 4 is ongoing for the duration of the use of the drug. As long as the drug exists, this phase 4 goes on. So how long this whole process continues? So like I said, it may take up to 15 years to come up with one successful drug molecule. So again, to recapitulate, so we have these are trials are done in 3 phases initial, and first phase is 20 to 100 patients, and this goes on for several months. Mainly the safety is the concern which is checked here, and the percentage of drugs which are suitably tested here are successfully tested in this case are 70%. Phase 2 about 100, which runs in hundreds, so which takes several months to 2 years, and it is for short-term safety, but mainly the effectiveness of the drug. So it's done in hundreds of people, and 33% of the drugs are successfully tested in this. And the phase 3 trials, it runs in thousands, up to 5,000 patients are involved here. It runs for 1 to 4 years, and the safety, effectiveness, and the dosage is the main concern in this. And about 25 to 30% remain successfully tested in this trial phase 3. So during this clinical trials, there is a thing called new drug application, NDA. So this is submitted to FDA if the phase 1, 2, and 3 trials indicate the drug is safe and effective. So if the drug is fine up to the phase 3, and the new drug application, because it's a new drug entity, experimental molecule, so and it is safe and effective, so this has to be submitted, the new drug application, and it is a comprehensive statement which includes the information related to the chemical structure of the drug, what was the basis, what is the scientific rationale for this drug, development, the purpose of the drug therapy, and the preclinical tests which were done on cell lines as well as the animals, and the laboratory results which were obtained, IC50 values, and all the clinical human testing data, drug formulation for the placebo as well as the active ingredient, and production details, how this drug was produced, and what was the proposed labeling, what would be the life of this drug, the expiry date, and the manufacturing to expiry, for example. And thousands of pages are being submitted to the regulatory agencies. Then the FDA has to conduct the preliminary review, and they have to decide if it is enough information, these thousands of pages, and to proceed further for the new drug application review. Then FDA requires to make a decision within 180 days of the date of the new drug application which is submitted. And FDA and the company can create mutual agreement to lengthen its time frame. If there is no enough data, they have to work longer time frames, and they have to substantiate all the required information related to safety and efficacy and the dosage. Then NDA decision making process can take up to months to several years, up to 7 years, but on an average about 2 years it takes. So it may require minor or major changes in the new drug application, and in addition to the clinical studies, and may inspect the testing, packaging facility, and ensuring the regulation. So this is a very important. So the factories are being inspected about the packaging, the testing, the cleanliness, and the kind of equipment which is being used, and the procedures and methodologies should be exactly accurate. It should be replicating each time the production goes. And then further, there is a post-marketing surveillance. Once the drug is released into the market, so we have to monitor the ongoing safety of the marketed drugs by reassessing the drug risk based on the new data collected from the after the drug is being marketed, and by recommending the ways of trying the most appropriately managed to manage the risk. It involves adverse reaction reporting if there are any adverse reaction in the long run. So the doctors have to report this appropriately in order to regulate the market of this drug. Periodic sampling and testing need to be done. Periodic inspections need to be done. What are the costs? So on an average, this successful billion-dollar drug, we so-call, out of the 10,000 molecules, the one which is successful can incur a cost up to $1 billion, 800 million to $1 billion each for each drug to be tested. The spending on clinical trials in the US is forecasted to rise by this year about 32 billion. And why would anyone spend that much money on the drug development? This is because of the profits. You can see here in the graph the Gilead Sciences, the Fabiflu, for example, the drug. So they can have a profit margin of about 50%. So huge profits. It can bring them a huge profit. Novartis, Apple, about 30. So these can get huge after the trials. They can get huge profit. That's why they want to invest onto these new drug